Bonjour tout le monde, merci d'être venu pour le deuxième séminaire de cet automne. On est très heureux d'avoir Sophie avec nous, une nouvelle membre de l'Église. Elle nous parle aujourd'hui des choses quand même intéressantes, de danse. Je trouve que c'est plus intéressant, c'est pour ça que je suis là. Comme petite introduction de Sophie, Sophie maintenant elle est professeure adjointe au département de biochimie, microbiologie et bioinformatique ici à Laval. Il a fait son doc à l'Université de Montréal sur caractérisation des formes fonctionnelles évolutives et la dynamique conformationnelle des bêta-lactonases.
Exactement.
Je suis là pour la danse, pas pour les protéines. Par la suite, j'ai fait un postdoc à l'Université de Duke où j'ai travaillé sur la méthodologie du biologie structurale et biophysique. Ensuite, j'ai fait un programme qui est, je pense, Duke Exploring Nuclear Medicine. Et ensuite, j'ai fait, c'était un autre postdoc. Un postdoc à Duke Human Vaccine Institute qui travaillait pas mal sur la caractérisation par microscopie électronique cryogénique, les effets structurels et antigéniques de nutrition dans les structures virales du Gobeil. Aujourd'hui, on va nous parler sur les protéines.
Merci beaucoup. Merci pour l'introduction. Merci tout le monde d'être là aujourd'hui. Pour essayer de peut-être être plus rapide, parce que mon français n'est pas encore à 100% revenu, je vais présenter en anglais, mais n'hésitez pas si vous avez des questions en français. C'est pas un problème. So today I'll be talking about the protein dance and why moves matter, why the moves that they do matter for their function. So, I mean, just to kind of introduce a little bit where I come from. So I did my PhD at University of Montreal under the supervision of Professor Joël Pelletier, where I was studying protein evolution and bacterial resistance using beta-lactamase. And then, so basically, it's kind of funny because I've evolved through my career at the same time as through the systems I was studying, but also the methodology for structural biology that I've used. So during my PhD, I use NMR, so nuclear magnetic resonance and X-ray crystallography. I then moved to antifungal drug design at Duke under the supervision of Professor Leonard Spicer. And then, well, like everyone, the COVID-19 pandemic hit pretty hard. And so I decided to move to the Duke Human Vaccine Institute and use my structural biology background to learn how to work with viral entry proteins. So we were working on the spike of SARS-CoV-2. And I learned to use a new tool, a new technique, cryo-electron microscopy, cryogenic electron microscopy, or cryo-EM. So you'll see throughout all of this, there was one question that I've always was really interested in. And it's, if you think of a protein that has a function, the vision that we have is often a lock and key, especially for enzymes. There's a lock, the key fits in, and then it's transformed. The substrate is transformed as to a substrate, a product. But if you think of a lock, there's more to it than just the lock. There's the whole mechanism inside where there's different pieces that have to move to adapt to the substrate or the key. So for protein, it's basically the same idea. You cannot just think of just a key fitting. There's also motions that are important for the function. And so this has been, it's part of the induced fit mechanism. So I'll talk about 3 different protein systems and how protein motions have been key for understanding what's really going on. So the first protein system I'll be talking is enzyme and how protein motions are important for catalysis or the function. And so I'll talk of beta-lactamase like this one. And then I'll talk about protein drug design and how protein motion can benefit designing new antifungal drugs. I'll talk about tacrolimus and this calcineurin protein. And finally, I'll talk about viral entry proteins and how motion can promote immune evasion or better entry into host cells. And I'll talk SARS-CoV-2 spike protein. So first, for the beta-lactamase story. So if I'm showing you this short clip of a person walking, and I ask you which motion is really the important one to fully describe what you're seeing, is it the legs moving? Is it the person going forward? Is it his heart? If you think of his heart, and if this person doesn't have one, I mean, it's not going to have the motion. So I don't know. And then if I'm showing you another clip like this one, and I ask you, does the horse fully become airborne at any point in this clip? To understand, to be able to respond to that question, you really have to take images at a specific time scale or frame frequency that will allow you to capture the motion you're interested in. The answer to that question is yes. And this was already shown in 1878 by Edward Medbridge that captured the horse that does become fully airborne. So for protein, it's basically the same idea. You have all kinds of different motions that are happening on different timescale. And depending on which question you're asking or what event you're interested in, you have to choose the right experiment to measure the protein motion that you're interested in. So for example, if you're interested in fast motion, you'll go for picosecond to nanosecond timescales. And the good news is that there's an NMR experiment that you can do to probe those motion. Same for ligand binding. If it's a little bit slower, we're talking mostly microsecond to millisecond timescale. There's also an NMR experiment to probe those motion. What's tricky is the motions that are in between. There's no obvious NMR experiment or easy experiment to probe those motions. So for this presentation, I'll show you an approach that we use using molecular dynamic simulation to really probe a continuum of motions through the system that we were studying. So the question I'm asking you here is, are protein motions conserved throughout evolution? To answer these questions, there's basically two parts to the question. First, are you talking about naturally evolved proteins? And so to study naturally evolved protein, we selected the beta-lactamase TEM1 and PSC4 that you can see here in blue and green. Those proteins share 40% sequence identity. And as you can see, they're highly similar in terms of their structure. So we probe the motions in those proteins on those three different timescales, so picosecond, nanosecond, which are fast motion. And I'll use a coloring scheme from yellow to red, which means less motion, more motion. As you can see in TEM1 and PSC4 on the fast time scale, so this is measured by NMR, it's motion mostly related to H bonds and side chain rotation. TEM1 and PSC4 are really similar, and the motions are mostly on exterior surface loops. So there's a general similar trend for those two proteins. On the intermediate timescale, so we measured this using molecular dynamic simulation. Again, you can see that the surface loops are the ones that are experiencing motions. And there's a general similar trend between TEM1 and PSC4, but we start to see what we call their dynamic personality. So for example, in TEM1, this little loop here is more dynamic than in PSC4. So it has a little bit of change, but overall, it's the same. And then we measured microsecond to millisecond timescale motions. So the motions that are slower and more related to the actual function of the protein, the turnover rate. And we can see that key active site residues. So the active site is right, sorry, maybe I should use my pointer, where there's the gray residues that are showing motions. And so overall, again, TEM1 and PSC4 are really similar one to the other one. So if we're asking the question, talking about naturally evolved proteins, over all the three timescales that we probe, TEM1 and PSC4, those two class A beta-lactamase, are generally rigid. They're similar, and there's a general trend of conservation of motion, which suggests that those are important for having good functional, fully evolved proteins. But then I told you there's a second question that comes with that main question conserved throughout evolution. What about artificially evolved proteins that would actually be representing evolutionary intermediate between TEM1 and PSC4? So how we did this is we created a bunch, well, actually, it's Francis Arnold created a bunch of chimeras that are hybrids between TEM1 and PSC4, where there's different recombination events that happen. So recombining different blocks from TEM1 and PSC4. And we think of those chimeras as potential intermediate on an evolutionary path that would start from TEM1 and going towards PSC4. So just to kind of present the system, we have TEM1 and PSC4, those two enzymes I've talked about that are fully evolved. And we have two different chimeras that I'll be talking about, CTEM17M and CTEM19M. So C comes from chimera, TEM, because it's closer to the TEM1 enzyme. 19M or 17M refers to the number of mutations that they're away from TEM1. So CTEM17M and CTEM19M have this same omega loop block that is exchanged from TEM1 to PSC4, and this introduced 17 mutation. So CTEM19M has two more mutation that comes from this serine 70 block that is exchanged. And all those mutations are really nearby the active site. So, I mean, the first question we ask is, are those enzymes still active? So just to kind of show the whole family portrait, we have TEM1, CTEM17M, plus two more mutation, we go to CTEM19M. To complete the square, we also made another chimera that we called CTEM2M, and this only incorporated that serine 70 block that is exchanged. Overall, all those proteins were really active. They were similar to TEM1. What's interesting is that we had a reduction in turnover rate for CTEM19M, and when we tested CTEM2M, we observed the same phenomenon, which suggests that those two mutations do impact slightly cephalosporin hydrolysis. So now that we know that they're active, well, did the recombination event somehow impact the protein motions? So we use the same three methodology to measure the three different time scales of motion. So this is TEM1 that I've showed you earlier. And as you can see on that fast time scale, all three chimeras are generally similar to TEM1. So that suggests that those motions are somehow significant, functionally important, and that's why they're maintained. So we then measured nanosecond to microsecond time scale using our molecular dynamic approach. And as you can see, again, the three chimera are really similar. And what we called our dynamic personality of TEM1 is now increased in all of our chimeras. So somehow maybe this is an important trait of TEM1 that allows it to evolve. But overall, the motions are really conserved. It's when we measured the motion on the slower timescale, the timescale of protein, the turnover of the substrate, that we were really surprised. So this is TEM1 that I've showed you previously. And this is the TEM2M. There was a lot of motion all over in the protein, all located in one of the domains. So the protein is actually two domains, and all the motions were only on one side of the protein. To our surprise, when we measured the motion in CTEM17M and CTEM19M, you can see that there is motion all over the protein. There were more than 60 different residues in each of those that were showing motions. And there was also what we called unassigned residues by NMR. And so those are residues that are moving on an even slower timescale that we're not able to probe by this methodology. So to take all of those together, we've basically shown that TEM1 and PSC4 conserve their motion. And those enzymes are fully evolved enzymes. They're enzymes that are, fully evolved. And so in our chimeras, what's interesting is that we maintain a general trend of fast dynamic, but we have an increase in slow dynamic, and they're all active. So we think that this basically hints at the promiscuity of the enzyme, and somehow the fact that the enzyme is able to adapt to new motion could result in better preparation in case of a new selective pressure, and eventually maybe promote survival of the bacteria because the enzyme is able to hydrolyze something new. And so now the question that I'm going to leave you on this, and I still don't have the answer to this, those enzymes are bacterial resistance enzyme. But if we're talking of a primary metabolism that is essential for survival and just basic function, do we know, do you think it would be as tolerant to protein motion change or not? I don't know. So this is the first part of today. Now I'll talk of drug design and how protein motions are important to design a new antifungal drug. So fungal infections are quite common. Usually they are superficial and not necessarily really challenging to treat. They're mostly skin or nails infections. It's when they become invasive that they are really challenging to treat, and it's especially through for immunocompromised population. And in most cases, those infections are actually associated with really high mortality rate and really low incidence of treatment efficacy. So there's a pathway that's been identified a while ago, calcineurin, which is a really interesting pathway for targeting for antifungal drug design, because it's a pathway that is linked to many of the different phenomena essential for the adaptation of the fungus to the human host. So virulence, high temperature growth. The challenge here is that calcineurin is also, well, it's targeted by FK506. The challenge is that it's also conserved in humans, meaning that if you want to treat somebody by targeting calcineurin, you would also hit the host. And as I said, most hosts who are having fungal infections are actually immunocompromised. So treating using calcineurin is not great. FK506 is a large macrocyclic drug, and it binds calcineurin in a groove formed between actually 3 proteins. So you have calcineurin A right here, and then one of the helics of calcineurin A is used to bind calcineurin B, and FKBP12, so a 12 kilodalton protein, is used as an adapter protein for FK506 to be able to bind to calcineurin. So the challenge is that FK506 or tacrolimus for humans is an immunosuppressive drug. So this is why it's not used currently as an antifungal drug, because you would immunosuppress a person who's already is already immunocompromised. So the first thing, because we were working with Aspergillus fumigatus, the first thing we asked is, well, here I'm showing you FK506, if you had it, you inhibit the growth of the fungus. But if you remove FKBP12, what you can see here is that the growth of the fungus is not inhibited. So FKBP12, this adapter protein, is absolutely necessary for FK506 efficacy. So because we were working on fumigatus, we decided to try to remove all the FKBP12 from the fungus and insert instead the human version of it and another fungus one. And to our surprise, what we found is that if we have the human version of the protein or the mucocystilloid ones, it doesn't inhibit the growth. FK506 is not potent anymore against Aspergillus fumigatus. So with different collaborators and within a group, we obtain crystal structures. So these are crystal structures of different fungal versions of the calcineurin complex bound to FKBP12 and FK506. And in gray, in all those images, you have the human version of that protein. And they share generally around 60% sequence identity, but as you can see, they're highly structurally similar. So that would be a challenge to figure out what makes the difference. We also obtained the crystal structure of both the apo form and the FK506 bound form of the FKBP12 protein. And comparing all the sequences of the different version of that protein, there was one key residue in that 80s loop, which is actually the loop that is right here, kind of closing on the drug. That was interesting because it was changing throughout the different organisms that we were studying. So we decided to use that residue, target that residue, and introduce the mutation where we, instead of having the human residue 88, we would have the fumigatus one, or instead of the mucor one, we would have the fumigatus one, right? And so we found that by mutating that residue, we were able to restore the activity of FK506 against in fumigatus. And we did the same opposite experiment where we took fumigatus and inserted the human version of this residue. And you can see now we don't have the inhibition anymore. So that means that residue 88 is really a key residue for being able for FK506 to be potent against Aspergillus fumigatus calcineurin. So as I said, I'm a structural biologist and biophysicist. So I needed to know how does this drug really binds in solution? What are the protein motions? So we measured the binding affinity using isothermal titration calorimetry. And we also use NMR spectroscopy to measure how the protein would respond to the binding event of the drugs to the protein. And basically what you can see here, so every single dot on that NMR spectrum basically means, so it goes from black to red. And so the black to red is like the change or the shift that you can see basically means that the residue is experiencing a different environment due to the drug binding. So if you plot the difference between the blue and the black and red here for every single protein, so that's the human, fumigatus, and the mucosal one, you can see that there's some residue that responds in the fungal proteins and not as much in the human one. For example, here the black one is the human one. There's residue that are impacted in the human protein, but not as much in the fungal proteins. And so this is basically plotting on the structure, which residue of each of those proteins respond to the binding of the drug. And you can see that the two fungal proteins are not the same as the human one. So that's good news. That means we could probably benefit from those differences to create a better FK506. We also use an approach in molecular dynamic simulation where we relax our crystal structures and just let the protein breathe and just observe the different contacts that were made in short simulations between the human form of the protein and the fungal one. As you can see, there's some residue that are kept throughout the 3 different proteins, but there's also differences. To kind of simplify this, we can also just plot it on the actual drug and the protein. So here, to make it simple, if you're above the green line right here, that means that the contact is more beneficial for the fungal proteins. And if you're below, it's more beneficial for the human proteins. So you can see that there's plenty of different residues, both on the drug and also on the protein side that can be targeted to be more beneficial for targeting the fungal protein. And as you can see here, there's a key residue, residue 88, that was pointed out in our study as being an important residue for having a more selective drug. So using this information, so we also measured motions on those proteins. And I told you, those proteins are really, really similar structurally. And we did measure the dynamic both on the human version and the fungal one. And as you can see, they don't behave the same way in solution. Every red spheres means that the residue is moving. And the mucosal has way more motions at 25 degrees compared to the human one. The FK506 drugs really quench all the motions in human protein, but still leave space for breathing in the fungal one. So we were working at the same time with synthetic chemistry group. And using our structure, what we found is that there's position here, C22, where is pointing out in the same direction as this residue 88 that we saw previously as being really important for the drug potency. So they modified FK506 and introduced an acetohydrazine group here to generate this new compound, APX879. So we obtained crystal structures, and as we expected, this acetohydrazine group was adopting different position depending on which of the proteins it was binding. So it didn't like the fungal version of the protein, while it really liked the histidine residue of the human protein. We relaxed the contact, and we think this is a really good candidate for now. Let me show you why this is particularly interesting. So we measured the efficacy of APX879. It does inhibit growth. It's not as potent as the FK506, but it's still, I mean, there's 30-fold reduction up to 130-fold for albicans. It's still binds less tightly to the proteins. We have a reduction of around 40 folds compared to FK506. But where we were really surprised is that there's a 71-fold reduction in interleukin-2 production, which means that the drug is around 70-fold less immunosuppressive. So the question here that we still don't have the answer is, well, it's a balance between potency and immunosuppression. So what would be enough for having this next drug that would be, I mean, the next antifungal drug that we would need? So with this part, I showed you that the key residue is really 88. It plays an essential role in the FK506, FKBP12 binding interface for binding to calcineurin. we identified key different contact between FKBP12 and FK5 and the different version of the protein. And I think this is my key message here. It's not because you have a protein that is really similar and has almost the same sequence that it means it's going to be the same protein. They can behave differently, even though you think they're the same. And really the power here is combining structural biology with biophysics and molecular genetics to fully understand how to target the best sites for having a new antifungal drugs. So again, protein motions. So now I'll talk of the viral entry protein of SARS-CoV-2, the spike protein. So the spike protein is on the surface of SARS-CoV-2. You can see here on this electron microscopy image. It forms kind of a crown, and this is why the name coronaviruses, corona comes from the Latin word for crown. The spike protein is a nomotrimeric protein. It's a glycoprotein, so there's glycans on there. And it has a lot of different motions, large domain motions. So the first motion is in its what we call pre-fusion conformation. The receptor binding domains, which are right at the center here, can exchange between a down state or receptor inaccessible state and then go up and become accessible for the ACE2 receptor and then promote the entry into host cells. But there's an even bigger change to the spike protein that we don't hear that often. It's the change between the pre-fusion conformation to the post-fusion conformation. So you can see here, it's a long filament, and this is what promotes the fusion between the host cells and the virus. This change between pre-fusion to post-fusion conformation can either happen in a spontaneous way. So basically the spike by itself shed what we call its S1 unit that contains the RBD, and a change between pre-fusion to post-fusion, or it can also be receptor mediated. So the ACE2 receptor pulls the RBD, and then the spike change from pre-fusion to post-fusion conformation. So in the news, and everywhere we hear about the spike protein more than the other proteins. Why? The spike protein is the protein that really promotes the entry of the virus into host cell. And what's interesting is also the protein that show the more drift between the different coronaviruses. That's where you have the most changes, even though the viruses are generally similar. And it's interesting that it's mostly also in the S1 unit. The S1 unit is the unit that contains the receptor binding domain and the N-terminal domain. And those two domains are the domains that are recognized by most antibodies to inhibit to neutralize SARS-CoV-2. So, I mean, there's other coronaviruses that have been around. Most of these are not, because slightly respiratory infections, and they're not generally a source of big concerns. But you might have remembered in 2002 and 2012, we've seen the SARS and MERS. We've been in this pandemic now way more than 2 years, over 2 years. And so I just want to give you a little bit the perspective as a structural biologist of how interesting it's been, but also stressful. So already by the end of 2018, there was report of pneumonia cases in China, and there was reports of viral, that it might have been a virus. So already by mid-January, there was cases that were reported outside of China, and there was also the sequence of this virus that was released. And so we realized that we were facing a coronavirus. At that same time, Jason McLennan at UT Austin and his group, so Daniel Warp, who's now a postdoc at the DHVI, they worked on producing the spike protein of SARS-CoV-2, transfection, purification, freezing the grids, and getting a structure. So within 11 days, they were already having their first SARS-CoV-2 spike structure that was done. And so they released by the end of February in PDB, but mid-February, the structure and published their manuscript in Science. And so you might remember also the virus and the disease changed their name. It was initially the nCoV-19, became SARS-CoV-2. The disease was named COVID-19. And I remember really the moment where my advisor, Pumbeta, came into the group and said, you know what, guys, we're working in HIV-1, and it's also a spike protein for HIV. And what about translating everything we know on how to work with the HIV-1 and protein, and now just help and work through what SARS-CoV-2 is going to do and how it's going to evolve. So that was a really good call from her. So I'll present different parts of how this thing has evolved. So first I want to come back to the first mutation, the D614G mutant, that was really one of the key ones that I think allowed everything else. And then I want to talk a little bit of the different variants of interest and variants of concern that we've seen, especially now Omicron, which has been not great. So D614G appeared in March, and by the end of April, it was everywhere. So you can see here, it started as the orange and then became blue, which is the G form everywhere. And there was a lot of reports saying, well, this virus is more infectious. We were working with Betty Korber and other groups, and we were trying to understand what does this mutation do to the spike. And as a structural biologist, and seeing that it was somehow fixed really quickly, it was obvious that it was doing something to the spike protein. It had to give the virus a benefit, some kind of benefit. So for kind of situating where the mutation is, it's in the SD2 domain, which is right before the furin cleavage site, which allows to shed this part, which is the S1 unit of the spike. So you have the NTD, N-terminal domain, the receptor binding domain. And you have right here in the S2 unit, what we call the fusion peptide, and this is what makes the spike fuse to the host cells. In most of the reports, you'll have also two mutations, two proline mutation of residue 986 and 987, right in between the HR1 and CH domain. For all the work we've done, we never included those mutations. So those mutations, I can talk more about these later if you're interested, but basically they were introduced because they stabilized the conformation, the pre-fusion conformation of the spike protein. And it was stated that if you remove those, the spike would change between prefusion to post-fusion. We found we didn't need it. So D614G, because it's in the SD2 domain, and there's a glycan right next to it, a lot of papers were suggesting that it might impact the glycans, or it wasn't obvious. So we use our cryo-EM workflow, and I just want to present what I mean by cryo-EM workflow. So we produce the spike protein in mammalian cell, purify using usual size exclusion chromatography and affinity purifications. And then we use a tiny little grid, which is probably 2 millimeters in diameter, and we plunge it in liquid ethane, which is really, really cold. Don't put your hands in there. And so using that grid, so we use liquid ethane because it's a vitrification process that is extremely fast, and it allows to form what we call vitreous ice instead of thick ice. And so if we have this kind of glass ice, we can see through, and when we place this tiny grid in this gigantic electron microscope, and extremely expensive, you can see on your grid the little squares. You can see they're squares, but in the squares, there's actually holes, and your protein is in the holes. So you can see here on my micrograph, all the dark gray spots that you can see are actually spike protein. And you can use different computational algorithm to take those images of the spike protein and rebuild a density map that you would use to see what the protein looked like. And then using different software, you can rebuild the model that would be fitting in your map. So when we did this for D614G, the first thing we saw is that the ratio between the 1 up and 3 down states, so the receptor inaccessible and receptor accessible conformation, was increased. We had 50-50, while for the D form, we had mostly 3 downs. But one thing we've seen in our data set is that there was a lot of heterogeneity in the maps. There was lots of disordered. How to resolve this? We actually made subpopulations of the RBD up and RBD down state. And then the disorder became less, there was less disorder in our map, and we could really see different state out the RBD and NTD domains were not exactly the same throughout the whole data set that we had for the up states. The challenge, though, is that you have a trimeric protein, and so meaning that you have 3 monomers for each of them. And we have now 10 different structures. So how do you compare those structures and make sure that you're really seeing differences? So there's there's differences that are obvious, like for example, this RBD up down, it's obvious that there's a difference. The entity, well, I mean, it's also kind of obvious that there's differences between those entities. But there's other domains, like for example, the SD1, where we didn't see any differences. So the question becomes, is it because of how we align the structures, or is it true that there's really no differences between all those SD2 domains? So to answer this, we use a methodology which is called difference distance matrices. And so basically what it does is that it takes every single protomer, every single monomer, and calculate distant matrices between every single pair of carbon alpha in the structure. And then you can subtract all those matrices, one from the other one, and basically get an image of your protein that is independent of how you superpose your structures. So basically, to interpret these, just know that if there's colors, that means differences. That's the simple way of looking at this. Here I'm comparing an RBD up versus an RBD down protomer, and you can see between the two structures, the RBDs are different. What struck our attention is that, as you can see, there's differences in the S1 unit, but the SD2 domain right here is really not allowing any change in the S2 subunit. And so we think that the SD2 domain is kind of acting like an anchor that blocks the propagation of all the motions from the S1 unit that are important for entry into the host cell, so that the fusion peptide and the fusion machinery is not broken or compromised. So another key aspect of the D614G mutation is that it lies right next to the furin cleavage site. So we use our system and introduce an HRV3C protease cleavage site to be able to control when the cleavage of the spike would happen, because it's an exogenous protein to the cell in which we express the spike. And so we measured how susceptible the D form or the G form of the spike would be to this HRV3C enzyme. And you can clearly see that the D form is not cleaved as easily as the G form. So really, we think the D614G mutation has a real importance for the cleavages of the spike and promoting the fusion mechanism. For sure, we ask, well, can we do the same with the furin? But it's a little bit more tricky, because now you have the enzyme at the same time as you express a protein, meaning that you have two kind of different type of spikes. So you have a spike that is not cleaved and spike that is partially cleaved. What is interesting, though, is that we did negative sign electron microscopy and SCM. And we could see that even if the spike was cleaved throughout the expression, it was still maintaining its trimeric folded kind of kite shape. So that was interesting. So I've shown you that D614G increased the protease cleavage at the S1, S2 junction, and that this SD2 domain is really an anchor that protects the fusion machinery from all the movements that are going on in the RBD and entity. So now I want to talk a little bit of the different variants, because there's been so many, and there's been so many mutations in those. First thing, I mean, all those mutations, they all increase binding to ACE2. They're all trying to make the spike protein recognizing better ACE2. And so most of these mutations are affinity enhancing, or they also impact binding of antibodies. Yeah, mutation in the RBD. So the first variant that we studied is the one that we call the Ming variant. So this variant was the one that was identified initially in Norway and Denmark, that led to the culling of millions of mink, because they were really scared that it would jump between the mink humans. It was actually an initial jump from human to mink, and they were scared that it would jump again back to humans. So we obtained a cryo-EM data set on that one, and we found that it was approximately 50-50 of up and downs. What was interesting is that we found a new state that we've never seen before, where the S1 unit, so this part here, which has the NTD and RBD, of one of the protomer in the trimer was completely missing. And so we think it's due to a mutation I692V, which is right next to the furin site. And it basically breaks a latch by disrupting a nitrogen bond. And so it makes the spike a little bit less stable. And so we use the DDM approach that I've showed previously, and we can see that the SD2 domain is not doing its role of an anchor as well as it used to do in, for example, the D614G variant. So that was an interesting one. And then we studied the alpha variant. So this is the variant that appeared in the UK in 2020. I don't remember exactly when. And this one was an interesting one as well, because it has a lot of mutations that were either destabilizing, for example, that T716I mutation that leads to the loss of a nitrogen bond, that were counterbalanced by other mutations that were stabilizing. For example, this introduction of an histidine triad that really stabilized the spike. And we found also another latch where there's a breakage of a nitrogen bond. So you can see here in the down protomer, it makes this hydrogen bond. If it goes up, normally this is broken, so it promotes the RBD and up from conformation. But now in this mutant, there's an A982 mutation, which basically breaks all the time this hydrogen bonds. And even more, there's another mutation kind of downward that pulls that loop down. And so that kind of increased the probability of the, that's probably why there's more RBD upstate for this one. There's a latch that is broken. What's interesting is that way before we've seen that mutant, that variant, we actually made a paper in an SMB where we use a really similar mechanism to control the conformation of the spike protein. We were actually breaking almost the same latch to stabilize an RBD up conformation. So it's interesting to see that, well, protein engineer used the same strategy as nature we figured out before. Woohoo, hooray for us. So then the beta variant, that's actually beta variant that kind of put two variants together. Those variants are the ones that appeared in South Africa and Brazil. They share a same set of 3 mutations in the RBD, which are what we call the infernal trio of mutations, K417N, E484K, and N501Y. Those, again, a lot of RBD up and a lot of disordered in our data set. So when we saw this, we started to wonder why antibodies are having such a hard time with those 3 mutations. And so we use a molecular dynamic simulation approach where we've seen that if you have the E484 residue in that, what we call hook for antibody latch, to latch on the RBD, usually if you have a E484, you have 50% of a hook conformation, which allows antibody binding or disordered state. But if it's mutated to a lysine, you can see now you have way more of the disordered state. So that's probably why antibody have such a hard time with the E484K mutation. And now it also increased the RBD up population because there's interactions between the 3 different RBDs on the spike that are broken. So it's easier to have RBD going up and be accessible to the receptor. So now I want to talk delta, which is the one that we've seen in India and was really bad as well. It was also associated with many fungal infections that were at the same time as this variant. So we solved a data set, 17 different structures of delta. Again, a lot of heterogeneity. What was interesting is that we found that same state that I've discussed earlier in the MINC for the first time in the delta data set. So basically, we think that this one, the different delta, because delta is also a lineage, there's many different forms of it. There's something going on with that SD2 domain and this P1681R or H mutation that really promotes better furin cleavage. We found also a reorganization of a linker that links the NTD and the RBD from a same proteomer that is kind of, it's rearranged, let's say it. And so there's less coordination between the RBD and NTD, and somehow we think it promotes the RBD to be able to go in up conformation and be ready for binding ACE2. So finally, Omicron. Well, this one. Omicron, when we were working with it, everybody had this, again, 2P mutation. And we were one of the few groups that decided to go not for the 2P mutation. It was good and bad. Extremely bad because we were getting maybe 5 micrograms per liter of expression, extremely painful to purify. But really good because we've seen that if you introduce the 2P mutation, the antibody binding, so for example, CR3022, which binds to the RBD, is impacted by the 2P mutations. So somehow those 2p mutations do limit the conformational diversity that we can see in our spike. What we found in the Omicron spike is that the spike is more compact. It's really tight compared to all the other spike that we studied. And we found again that N2R linker that was shifted, so probably more RBD, more ease to go in the RBD up conformation. At the same time, I was working on a new antibody, TH1058, and this is an antibody that binds to the fusion peptide of the spike. This is a really interesting site because it's conserved throughout all the different variants. There's almost no mutation in that region. And what we found is that Omicron, the TH1058 antibody was binding much better compared to any of the other variants. And so the first question we asked, well, is it possible somehow there's an increased affinity for it? Well, it seems not. The SPR binding data showed that TH1058 binds as well to any of the variants. As expected, we produce also spike that are cleaved. So the dashed line are cleaved spike at different site. If you cleave the spike there, the fusion peptide becomes more accessible. So using crystal structures, it's interesting because I was working on something else and I got that crystals and everything just came together. We got a crystal structure of the H1058. So this is the heavy chain and light chain. And here we have a small peptide that mimics the fusion peptide of SARS-CoV-2. And we use our cryo-EM reconstruction of Omicron, where we could see that the fusion peptide was more defined than any of the other structures that we had. And we dock the H1058 to the cryo-EM reconstruction, and it was just not working out. Basically, the antibody is making too many clashes with the prefusion conformation. And so that says that the Omicron spike has to have his fusion peptide more accessible or having more motions or more dynamic so that this antibody is able to bind so much better to Omicron. So with this, I mean, what I've shown you is that all those variants that we've seen, and probably the next one to come, they all have different mechanisms to come up to the same event. They just want to have more RBD in the up conformation. And so what we've seen is that different set of mutations are all able to do this. So, I mean, what's next? So I kind of wanted to also quickly just hit on what I want to do now because, well, I'm a new member of the ABIS. So I showed you different mutations in immunogenic regions, and I've showed you that there's differences in our computational analysis of the RBD, especially for the E484K mutation. So now I'm interested in using NMR to actually look at the RBD and see if some of those motion, well, do they really promote immune evasion? Are they important for antibody binding? Or are they important also for ACE2 binding? And can we engineer ACE2 to kind of make what we call decoy type receptors, so we could use it as a therapeutic way of treating against SARS-CoV-2. There's different antibodies and different coronavirus also that now binds to ACE2. So our protein motion is important for the interaction. Another, I've mentioned for Delta, there was a lot of cases that were reported of fungal infection associated to the COVID-19, the Delta variant. And so I want to study if opportunistic infections, if somehow Delta promotes opportunistic infection. So there's been reports of many different receptor or co-receptor, or there's so many different names for these that could be helping out the entry of the virus into host cell. So using my structural biology approach, I'm curious to go see if I can capture those interactions and understand if somehow they promote opportunistic infections, fungal infections mostly. Another, well, this is maybe more for the future. I'm interested in fungal and bacterial co-growth experiments where sometimes if you have two different things growing together, you would start to have kind of a bacterial fungal warfare going on. And so you would start to produce new compounds. And so using, there's a new tool called cryo-IT. You can use basically cell extract and try to figure out what are the proteins that are now induced if you have the two things growing together. So this is something I'm really interested in to try to figure out if you can have better drugs. So with this, I want to acknowledge the groups with whom I've worked, all the funding sources. I mean, this was really important, especially in the pandemic time. This is my lab website, really active on Twitter. And yes, I'll be recruiting students in 2023. So this is a message for anybody who's interested. Don't hesitate to send an email for questions about the project, and I'll be happy to take any question you might have.